The eukaryotic cell is often viewed as the biological baseline, featuring a highly compartmentalized interior with distinct physical structures. Prokaryotic means before the nucleus, representing an older lineage. Next to a eukaryotic baseline, an average bacterial cell is 10 to 100 times smaller. This physical size disparity points to an internal architecture built on a separate set of rules. Microbiology at this scale requires discarding the animal cell template. Scaling down a complex organism does not account for the specific mechanics of a bacterium. A common analytical trap is the assumption that a lack of a nucleus implies a lack of genetic material. Bacteria hold large amounts of DNA, but utilize a storage strategy that excludes a surrounding envelope. A prokaryote lacks the specialized internal structures of animal or plant cells. It contains no mitochondria, no chloroplasts, and no internal membranes. In this domain of life, no nucleus describes the absence of DNA packaging rather than a lack of the genetic code itself. A bacterium operates on an open floor plan. All genetic, structural, and synthetic components share a single, continuous environment. We can construct the prokaryotic model from first principles, starting with an empty cytoplasmic void. All cellular life requires a boundary. Here, it is the cell membrane. This membrane mediates the transfer of materials into and out of the cell. Within the cytoplasmic space, we find the ribosomes. These are the dedicated sites of protein synthesis, reading genetic instructions to build the molecules required for survival. While bacterial ribosomes are physically smaller than those found in human cells, their core mechanical operation is the same. These two components, the membrane and the ribosome, represent the universal biological baseline shared across both the prokaryotic and eukaryotic domains. The next structures are distinctly prokaryotic, beginning with their primary method of genetic storage. This is the chromosomal DNA. This is a single, continuous, unbroken loop, distinct from the linear, disconnected chromosomes found in human cells. This loop contains the foundational genes necessary for the cell's basic survival and reproduction. This vital code floats unprotected by any internal membrane. Lacking a nuclear envelope places the chromosomal DNA in direct contact with the ribosomes, an arrangement that allows for rapid protein production. Bacterial genetic architecture is bipartite, relying on a secondary modular system operating alongside the main chromosome. These are plasmid DNA. Plasmids are autonomous genetic modules, kept physically separate from the main chromosome. They carry bonus genetic traits that provide situational advantages in specific environments, such as the ability to survive in the presence of toxins. A single bacterial cell can hold multiple copies of these small plasmid loops simultaneously. For interaction with the environment, bacteria utilize flagella. Flagella operate as external motility engines. These long structures rotate to physically propel the cell through its environment. The combination of auxiliary plasmids and flagellar propulsion provides the adaptability and mobility that define the bacterial survival strategy. We can now view the fully constructed bacterial cell as a complete system. All 5 major cellular components operate simultaneously within this single space. The chromosomal DNA, the plasmids, and the flagella identify this domain of life. Prokaryotic efficiency stems from genetic material that floats freely in an open environment. This open floor plan creates a streamlined architecture optimized for rapid reaction. It enables the bacterium's most effective survival mechanism. This microscale cellular anatomy has direct consequences for the medical crisis of multi-drug-resistant superbugs. The plasmid is the primary vehicle driving this crisis. Because plasmids are separate from the main chromosome, they can be copied and exported out of the cell through horizontal gene transfer. Bacteria can pass these genetic modules directly to neighboring cells, bypassing the timelines of normal reproduction. The most critical trait carried by these transferring plasmids is the genetic code for antibiotic resistance. This allows a single resistant survivor to distribute its protective code through an entire population, or even across different bacterial species. The uncompartmentalized modular architecture of a bacterium is the physical mechanism that facilitates this rapid genetic adaptation. It remains one of the most resilient biological designs on Earth.